Hello, microbiology students. This is your instructor, Miss Wahardo, and we are going to be going over our first lecture, Lecture 1, which covers Chapter 1. And this is the main themes of microbiology from our text Tularo's Foundations in Microbiology from the 11th edition. So without further ado, let's get started. Microbiology. What is it? Well, it's the study, an area of biology that deals with living things, ordinarily too small to ever have been seen without magnification. And this is a very broad and large area of biology that includes bacteria, archaea, fungi, protozoans, and viruses. And before we can delve into those specific groups, I'd like us to go over the history of microbiology. And I know this is a little out of order from your textbook, but I think it's better explained in this order. And so, prior to the 1600s, right, the microscopic world was not yet explored. And it wasn't because people were against it, they just didn't know about it yet. And so people understood that there were unforeseen forces at play that were causing things such as illness and disease. However, due to the lack of technology, they really couldn't explain why such phenomena was even occurring in the first place, whether it be superstition or otherwise. Many people believed in this concept called spontaneous generation. And so, what is that? Well, it's the belief that living creatures could arise from non-living matter, and that such processes were commonplace and just regular. So, to give you a better understanding of that, um, these people, they believed that life could arise from dead matter or decaying animals outside. And so they would notice and observe that maggots and, like gnats and flies, they'd appear out of rotting meat and corpses. And, they didn't think at all that, these flies were laying their eggs in this rotting meat. No, they thought that they were spontaneously generated from this dead material. And so that's how they believe, that's how some early people believed life came about. And so we move on, and we get Antony von Leeuwenhoek, right, in the 1600s. And so this man, he was originally a Dutch linen merchant, right, but later becomes a self-made microbiologist. And how did that happen? Well, he developed lenses in his free time after work at his shop, and he would view the thread counts using these incredible lenses. But from them, he became increasingly more curious about his surrounding world, and so he would look at various other things through his lenses that he had built. I believe your text says over 250 microscopes to look at, to look through. And one of the first things that he observed from his little microscope was really old rainwater that had been stagnant in a clay pot in his house. And he looked through it, and he noticed that there was, a whole little world just thriving with these tiny, tiny organisms, which he called little living animalcules, or little animals if you translate from Dutch. And so he had said that in a single drop of water, that all the water seemed to be alive. And so from that, his curiosity grew, and he was probably the first person to have ever observed red blood cells. He'd also observed sperm and tartar from people's teeth. He was looking at everything and anything under these scopes. And so here are what his microscopes look like as we had looked like previously in lab. And another incredible thing about Leeuwenhoek is that he would document everything he saw. So there are journals just filled with everything he had seen, and you can even guess what exactly he was looking at, what particular species of microorganisms. And so this is just a replication of what could have been seen in his journal that your text included on figure 1. And so people were still believing in that theory of spontaneous generation. Even with Leeuwenhoek's discoveries, it was still very hard for people to wrap their heads around that, there was a whole other world that could be causing illness and infection. And with that, let's start talking about an incredibly devastating disease. And so this is smallpox. And smallpox was a death bringer. It was a horrible, horrible illness that was caused by the variola virus. And this virus has been in existence for over 3,000 years. And how do we know that? Well, you can find scars and old pustules on mummies. And that's how we're able to date this incredibly deadly disease. And so not only was it very deadly, it was horribly contagious. And so you could contract smallpox from, the you could aerosol. You could get it from, droplets in people's saliva. You could get it from just skin to skin contact of someone with pustules. And 3 out of 10 people who had this disease would die. It caused millions and millions of deaths. Survivors were able to, live past smallpox. They were left with horribly disfiguring scars, and they were in pain because this not only just left, rashes on the surface, and pustules on the surface of the skin, it was doing this internally as well. So these people were incredibly, incredibly agonizing in pain. And the sad thing about smallpox is that there's no treatment or cure for it. And so the best they could do was this method called variolation. And variolation, coined after the name variola virus, it was a method that basically exposed patients to smallpox, and they do this by using scrapings from a smallpox pustule on an infected person. And then they would either scratch it into a patient's arm or have the patient inhale the scrapings through the nose. And the patient would still contract smallpox and they would still develop symptoms. However, this was a more mild and left less scars than if they had acquired this disease naturally, so to speak. So Edward Jenner, with variolation in mind, he discovers vaccination. He's the creator of vaccines, and he also is what some would like to say the founder of the scientific method because he developed a methodical approach to organize an experiment that had repeatable results. So, how did he come about this? Well, he had noticed that cows were exhibiting a pox similar to that of smallpox, and he had realized that milkmaids um, often would get lesions and they would contract the pox from the cows. However, these lesions would only go up to their arms. But that wasn't the most notable part. He noticed that these milkmaids were immune from smallpox. And so that was his observation. So he raised the hypothesis that, if you were to be infected with cowpox first, uh, would you be immune from smallpox? And so what he did was he inoculated a boy with cowpox, as you would have, during variolation. What he would do is he takes scrapings from the pustule and he put it into the boy's arm, and he exposed the boy twice to smallpox, and he never contracted it. He was immune. And so he repeated these results on 23 other people, and then this got the attention worldwide. allowing for vaccines for smallpox to become commonplace. This was revolutionary. People were just astounded by how quickly and amazingly they were able to prevent such a deadly and horrible disease. And so today, smallpox has been eradicated. The last known person to have ever died of smallpox was in 1978. It was a woman, and she passed away from it within a year of contracting it. I think it was a little less, actually. And it's actually speculated that she didn't catch it, um, you know, from someone out in the outside world. She got it because they were doing a study in a microbiology lab in that hospital, and so many have come to the understanding that she contracted smallpox from the vents. So, guys, it's really good to have a very safe lab procedure. This was a very sad thing that occurred. And so, moving on from Edward Jenner, we go into Louis Pasteur. And so, he was around in the late 1800s. And who was he? He was a renowned chemist. He was around a renowned, scientist in general. He was from France, and he was asked to help. Because he was so well known, he was asked to help with problems with the alcohol production in France, and they were just doing horrible in production. And so with that, he tackled on this study, and so he had discovered that some microorganisms were able to survive without the presence of oxygen, which we know today as anaerobes. And so he realized that these microorganisms underwent different processes than aerobes did. He realized that they'd go undergo fermentation. And so from that, he realized that, they would produce different gases. And so you could produce wine and beer by using them to ferment things. But he also learned, right, that heating up liquids could kill microbes that you didn't want. So this was known as pasteurization. He did a lot of studies using lactic acid. So sometimes when you think of pasteurization, you don't immediately think of wine and beer. You think of milk. But that's because he worked a lot with lactic acid. Another amazing and incredible thing that he had done to the world of science and to the medical world was create vaccines for anthrax and rabies. Um, it was kind of sad. Your textbook includes that he had first tried the vaccine on a small boy, and he had inoculated him with the rabies virus, and he didn't know if it would work, but it did, luckily. But what a gamble. um, wouldn't happen in today's time for sure. Next, we have Robert Koch, and he was in the same time period as Pasteur. In fact, they were rivals, and so they both often infringed on each other's work. But because of that, we have an incredible amount of advancements in medical microbiology. And so, what he's most known for are his postulates, or Koch's postulates. And so, basically, he had posed these questions or criteria, and they would help to determine if a particular microbe was pathogenic or causing. And so, this helped to determine which microbes also caused what diseases. So, he was able to not only tell if a microbe was causing a disease. He could figure out which ones were causing what disease. And so these postulates that he created helped to further support the germ theory of disease, which is just the theory that states that microorganisms can cause disease, which, today is commonplace, but back in those times, it was something kind of mind boggling. So with his postulates, he was able to further state this theory. He discovered things such as tuberculosis and the causative agents for anthrax and cholera. And he and his colleagues also invented many techniques that we use today in microbiology. And he used these techniques for isolation, preparing media, maintenance of pure cultures, and how to prepare specimens for view under the microscope. And before them, right, we have Dr. Semmelweis, right? And what did he do? He introduced the concept of hand washing in hospitals. And that might seem mind boggling to us to think, wow, they weren't washing their hands in the hospitals. Uh, but it's just not something that people had thought about. I mean, only 200 years ago they were thinking about, microorganisms even existing. And so he had noticed that women in the maternity ward were not doing well. He wanted to know why. He had observed that women were experiencing a lot of infections, especially after examinations from physicians. And these physicians had just come from places like the autopsy room, and he realized and put together two and two, which to us today wouldn't be a big deal, but in these times it was. But he realized that hand washing prevented the passage of bacteria between people. And so, they realized, wow, these small microorganisms can also be on our hands. And so there was a lot less complications in the maternity ward because of this, a lot less infection, and better life overall for these mothers. Um, Another incredibly important person in the world of microbiology is Joseph Lister. And so he came after Semmelweis, but using his ideas and concepts, he developed what we know today as aseptic techniques. And he used these for surgery. He realized and applied those techniques of, keeping a clean area to the operating room. And so he applied hand washing and chemical sterilization using incredibly strong chemicals such as phenol. And he would heat sterilize the tools that they would use for the operations. And then he'd also soak bandages in phenol, and so this prevented further infection from his patients. And so there was better survival rates and, better overall healing of the wounds compared to those of people who did not practice aseptic techniques. And so now that we went through the history of the study of microbiology, let's get into the actual subjects of microbiology, which is the microorganisms themselves. So, well, let's begin with the start of the Earth. Earth is estimated to have formed about 4.6 billion years ago. The first living organisms, well, they arose at 3.5 billion years ago. But I think it's fair to note that 3.5 billion years ago is just an approximation from the fossil record, because it's an approximation because there is a little bit of bias in the fossil record in that soft-bodied organisms, they don't preserve well, and so there's a lot of limitations to them being able to be preserved in rock and such, just because they're too delicate to do so. So for all we know is that these organisms could have been even a little bit earlier. They could have been around earlier. And so, what were these organisms? They were prokaryotes. Prokaryotes, right? That means before the nucleus. Pro meaning before, karyote meaning nucleus. And karyote is Greek. It comes from the Greek word karyon, which means a small nut, a small kernel, or a package. Take it as you will. I think that's a cute note to note. But prokaryotes, they dominated Earth for over 2 billion years. They were the only things present, and they actually prepped the Earth for future life to be there. And so, because of that, around 1.8 billion years ago, more complex single-celled organisms start to appear in the fossil record. And these slightly more complex organisms, they're referred to as eukaryotes, meaning true nucleus. And these were single-celled organisms that contain membrane-bound genetic material, the nucleus, and membrane-bound organelles. And so these early eukaryotes, they most resembled simple algae and protozoans. Um, but from these eukaryotes, right, these early eukaryotes, they give rise eventually to multicellular organisms such as fungi, plants, animals, meaning you and I. Thanks, eukaryotes. So let's go ahead and just kind of visualize how long, you know, our microorganisms dominated the world. And so, we have at 4.6 billion years ago, that's the origin of the Earth. 3.5 billion years ago, this is where life begins. And they have stayed and maintained life up until present day. And it's incredibly impressive the amount of time that they've had to evolve and advance over those times, and how much they were able to contribute to, other life on Earth. Without them, we wouldn't have, oxygen or, habitable places for us to live in. And if you can't really wrap your mind around that or get too excited about it, let's get excited. So, if the evolution of Earth, right, was scaled down to a 24 time frame, right, life begins, our little prokaryotes, it begins at 4 a.m., 4 in the morning, right? And it's just these guys all the way up until I'd say around 1 to 2 p.m. That's when we get the eukaryotes, right? And eventually we get so algae and stuff like that, right? And so that's not until the p.m., right? 4 a.m. all the way until 1 a.m. And you're probably wondering, well, what does that mean about us? When do we arrive? How long have humans been on Earth if we're doing a 24-hour clock? We've only been around long enough to last, to basically have existed for the last minute and 17 seconds. And that's for our ancestors. Modern humans don't arrive until the very last second. And I think that's incredibly mind-boggling to think of how how late we are to the party and how long the Earth was dominated by these microorganisms. And so, thinking of these different organisms, we're going to get into taxonomy. And what is taxonomy? It's the formal system for organizing, classifying, and naming living things. And so, when you refer to a particular organism, it's difficult to do so when there's multiple different names of these organisms from multiple different people studying that same organism. And so, because of that, we do use the Linnaean system to this day. What is the Linnaean system? Well, it was a hierarchical organizational system developed by Carl Linnaeus, who was a Swedish botanist, and he was tired of all this confusion he was having to going through different scientific meetings and meeting with other scholars, trying to just cipher through and figure out if they were working on the same organism or not. So in his book Systema Naturae, the 10th edition in 1756, he explains the Linnaean system in which to name and categorize life on Earth. And so, looking at this hierarchy, right, we have main taxa in a classification scheme, and we have them organized into different descending ranks, right? And so you start off very large, and then you get to something much more specific. So you start with the domain, right, which is the broadest description of things, And so if we go through it using this red fox as an example, right, the domain would be that they're eukaryotes, right? So they have a true nucleus. Kingdom would be that they're animals, right? They're heterotrophs. They're multicellular. Okay, still very vague, very broad. Then we say, guess what? They've got a nerve cord, so that's chordata, right? But let's get a little more specialized than that. They have fur and mammary glands, so they're mammals, right? A little further specialized, they're carnivores. They eat meat, right? They have that in their diet. But let's get even further than that. And guess what? They have canines, so they're canids, right? They're dogs. Further than that, they're foxes, genus Vulpes. Species, right? The as specific as we can get, it would be Vulpes vulpes. That's the red fox. And so your textbook has this on figure 1.12 on page 20. And so it gives you these two different examples, right? On one side, we have us, right? Little Homo sapien baby. And then we have the Paramecium caudatum, right? And so just giving us further practice with this, right? Let's start with the domain, right? These are both eukarya, right? They're all eukaryotic organisms. But guess what? All these guys are animals, which is very vague, right? But so we want to get further than that. So guess what? They have a nervous system, a central nerve cord. So goodbye, sea star. You do not have a nervous system. Out of here. Let's get further than that. We go into the mammals, right? Get rid of the sea squirts. They're gone. Goodbye, Urochordata. And then we get even further specialized to that. We go to the order Primates. Goodbye, dog. You are a canine. Further into that, we get into the family Hominidae. They they put Hominoidea, which is superfamily. That's wrong. It's the family Hominidae, and so that's the great apes. But let's get even further, and so here is the genus Homo. But there are other organisms in the genus Homo. Well, there were. They're extinct now. What are we? We're sapiens, modern humans, Homo sapiens, right? And so you could do that with this side as well. We start off with kingdom protista, right? And so this is all protists, which includes the protozoans and the algae. But let's get even further than that. Guess what? They've got cilia. So this is Ciliophora. And so we have these different organisms, but hey, we really, really want to get more organized than that. We want to keep as much vagueness out of here as possible. So let's go into Oligohymenophora, right? These are rapidly swimming cells. They're single cells, and they have rows of cilia, and they have ciliated oral grooves. But we still want to dive further. So we go into the order Paniculata. These are dense cilia spread all over the cell, and they're paniculae, trichocysts there in the outer membrane. But let's get even further. Family Parameciidae. And then even further from that, Paramecium, species Caudatum. Let's go. So, not only do we use hierarchical classification schemes, but we also use a binomial nomenclature system. And so what is binomial nomenclature? Well, that's using two names to identify every organism. And so as you can see up over here, right, we have Homo, which is the genus, that's the first name. And then we have sapiens, that's the second name, which is a specific epithet. And then we have over here, right, the genus Paramecium, specific epithet, Caudatum. So together you make the scientific name. That's Paramecium caudatum. That's Homo sapiens. And so to generate these names, they come from Latin or Greek words. However, if the name comes from something other than Latin or Greek, the endings are revised to have a Latin ending. And so names can be created using the geography or where the organism was found. Or you could even do it from the morphology. You're describing what the organism looks like. And then we have uh, the name of a person, and that can be used to generate a name. And so your textbook has some images of those that you can look back on. But one of my favorites would be the American Robin. And so that one's funny. It's Turtis. Oh my goodness. But that's a little descriptive. But let's go on to understanding why we classify these organisms. Um, when we do that, right, we have to understand their phylogeny. And so, what is phylogeny? Well, that just describes the evolutionary history and relationships that are among or within different groups of organisms. So, we're just trying to understand how they're related and how we would place them into these classification schemes. And we can't do that without the evolutionary history. And so what is evolution? Well, it's very simple. It's just a change in allelic frequencies over time. And so what does that mean? Well, it just means that over time an allele is favored over the other, and so we end up with these different traits over time, right? They're displayed because there's a difference in alleles. And so the fossil record, the study of morphology, physiology, and genetics can help us um, to support evolution, to show that it exists. But because of it, we're able to better group these organisms. We're able to paint a better picture, a better map to understand how all of these organisms can be organized and how they can be related to one another. And so typically, people will organize them into 5 groups. And so your textbook, this is the figure they have, but as you can see, this is how we would organize, right, the phylogeny of life. And so at the base are very our oldest groups, right? They're the ones that have maintained these primitive characteristics. But as we go through, we have these different branches of life. We have the protists, right? Which go into our our eukaryotes, right? That's the start of the eukaryotes, right? We started with these prokaryotes, but then over here we start to get new characteristics, right? At the branch, we get a eukaryote. So right here is where we would begin eukaryotes. And eventually those branch out into plants, right? Um, and eventually into fungi and animals. And so we're able to just organize these different forms of life using, right, this evolutionary history. However, for this course, right, we could talk all day about all kinds of life forms, but for this course alone, we're just going to focus on the microscopic side. And we're going to focus on the groups bacteria, fungi, algae, viruses, protozoa, and the helminths, right? And with that, it's good to think about the characteristics of these organisms. And so there's 2 basic cell types from these groups, which, as we had discussed quite a bit in this talk, was prokaryotes and eukaryotes. And so, our prokaryotes, right? They're the ones lacking in a nucleus, right? Our eukaryotes have a nucleus over here. We just have, a ring of our genetic material, right? It's not encapsulated in a membrane. But over here it is, and guess what? We also have membrane-bound organelles, right? Other membrane-bound organelles. And then there's also viruses, which we don't really group as a cell. They're kind of their own little odd group, which we'll get further into in this course, which I think is going to be pretty great. So for the next class, we're going to go into finer detail over eukaryotic cells, and I can't wait to see you then.